Okay, so you're working on your nervous system. You've got Connect assignments, you have Learn Smart, you have homework for chapters 12, 13, 14, and 15. That's 4 chapters. That can be kind of intimidating, especially because y'all are getting sort of on the verge of exhaustion. You're not there yet. You may think you are, but you're not. Just wait. But bear in mind that while we're covering chapter 12 very thoroughly, when we get to 13 and 14, which is the central nervous system and the peripheral nervous system, we're going to skip a lot of that. Do you remember when we did joints and I said you don't have to learn any of the specific joints? And when we did muscles and I said even though there are 700 of them, we're only assigning you basically 70 lefts and rights. So 35 assigned named muscles. So likewise, when we get to the brain, are you going to have to learn all the parts of the brain? No. All the parts of the spinal nerves and all their branches and where they go? No, we're not doing them all. Okay, so don't panic yet. Okay? If you're looking ahead at those, don't panic yet. All right, so just bear with us. We'll get through this. What we talked about at the end of class last time was we had gotten through the physiology of how the dendrites do their thing with local potentials, how the axon carries the action potential, and we got as far as the synapse, and we've already met the synapse. We met it with neuromuscular junction, so this was really kind of a review of what happens. The neurotransmitter secreted, it binds to its receptor, which if that's a direct receptor, remember that means the receptor is part of the channel itself. It binds directly to the channel and opens it. And there are others which we don't even have pictures of yet, where it's indirect. And I sort of drew you a little picture of that. And then we kind of looked at a reflex arc, which you're doing in lab today, right? Okay. So I'm going to leave the details of what do you mean reflex arc for when you get to lab. We were looking at this from the standpoint of what is happening, where are the synapses, and what's happening. So as you get to the first cell that's being activated, it's here at the skin receptors, right? So remember what happens there. If those are mechanoreceptors, tactile receptors, the little channel's open because it's stretched or pulled or prodded or vibrated or whatever, and that gets you a local potential. And if that local potential is a threshold potential, what happens next? Then you get an action potential that zooms down the axon all the way to the synapse, and of course what happens when it gets to the synapse? Gates open, neurotransmitters release, neurotransmitter binds to the dendrites of the next neuron. And if it's sufficiently strong stimulus, then it reaches threshold. And if it reaches threshold, then what? Then you get an action potential down the next axon to the next synapse. Is that reasonably clear to everybody what's going to happen? Okay, what happens if it's subthreshold? What happens out here at the nerve ending if your stimulus is not quite strong enough? If it's subthreshold, you don't send an action potential. It's that straightforward, isn't it? So has that ever happened to you? Subthreshold stimuli? That's kind of a trick question. Are you aware of subthreshold stimuli? No, you're flat not aware of them, right? But have you ever looked down and saw a mosquito feasting on you and you didn't feel it? You didn't know it was there till you looked at it? Makes you say evil words that you don't want your kids overhearing, right? Okay, so that's subthreshold stimulation. Those mosquitoes are pretty tricky little things. And very often they manage to land so lightly that they don't stimulate even the most sensitive of the tactile receptors in your skin. And that's how they get away with that. If they couldn't do that, they wouldn't still be around because we'd have smacked them all and extincted the species. Okay, questions about that idea? Okay, well, we're moving in a direction towards understanding summation. And I mentioned this last time, but I didn't dwell on it because I knew I needed you to have a fresh brain before we really got into it. So summation is simply adding together the effects of stimulation at multiple synapses. And you can have a bunch of synapses all sending signals at the same time, or you can have a bunch of, or a smaller number of synapses that fire one after another. So we have spatial summation and temporal summation. This is very similar to what you did or what you were supposed to do in lab. We talked about it in here You didn't get to do that lab, but we talked about what happens if you increase the frequency of stimulation versus increasing the voltage of stimulation. What happened to your muscles when you increase the frequency? They just went faster, right? But if you increase the voltage and you give stronger electrical surges, it increases the number of motor units contracting and you get a stronger contraction, okay? So it's similar to that, spatial and temporal. You can change how strongly you're stimulating at one time by stimulating multiple synapses all at the same time over distance, over space. That's spatial. Or you could take the same axon and just have it fire repeatedly, and it would release neurotransmitters so rapidly that it would create a stronger stimulation. So basically what integration is putting that whole idea together. And I'm going to draw you, let's come back to that. I'm going to draw a little picture of that for you so that you can get some sense of how integration works. So I am going to focus in on a part of the spinal cord that is stimulating some motor nerves that are going to go out to withdrawal reflex to pull my hand away from a hot stove. Are you with me on that? Okay, so what I've got to look at is all of the neurons that are coming in from the pain receptors and the heat receptors in my skin, down from the brain. I may have different ones coming down in from the brain, and all of these things are going to impinge on my little association neuron. Are you with me so far? Okay, so what would happen, all other things being equal, if I touched a hot stove and I got this really strong response from my sensory neuron saying, boy, that's hot. What are you going to do? You're going to withdraw, right? So you're going to send that impulse right on through to the motor neurons, boom, withdraw before you even think about it, right? That's a reflex. You're not thinking about it. Okay? And that's what will happen if you're not thinking about it. But what if you had a pot of spaghetti boiling over on the stove? You see it, you're not touching it. Okay? Is that going to trigger your reflex? No. But it may prompt you to go grab that pot to pull it off the stove. Are you with me so far? So I have impulses coming down from the brain saying, that's a hot pot, I better take it off the stove. But what happens as soon as you grab it? You didn't think to have a pot holder handy. And it was a cheap pot, all metal. So when you grab it, what happens? That impulse is coming in from your sensory system saying, you need to let go of that. But what is your brain saying? No, it's going to make a mess. I can't let go of that pot. Okay? Or you'll get burned worse, or you'll spill hot boiling water all over you, slimy pasta all down the front of you. Your 2-year-old is pulling at your leg. You know, all of these things are going to be providing input. So what does this poor little association neuron do? Is it sitting there going, I don't know what to do? It's not doing that at all, okay? So what's happening really is that at the axon hillock of that association neuron, here he is in close-up. He's got all these multipolar dendrites. Here's the hillock. Here's the axon, okay? At any one moment, that hillock here is either subthreshold, threshold, or suprathreshold. That's the only choices. It's either at threshold, below threshold, or above threshold. There's really no other mathematical option. If it's at threshold, what does it do? It sends an impulse, which causes the contraction of the muscles, which initiates the withdrawal reflex. Okay? If it's below threshold, it doesn't send an impulse. It may be excited, but subthreshold excited. What if it's suprathreshold? It sends repeated signals. That's exactly what it does. So it sends repeated signals, and so you move a little faster or a little more strongly. Okay? So how strongly that reflex is activated, how quickly it's activated, depends on the speed and the frequency of action potentials from that association neuron. Okay? So looking at the dendrites of the association neuron, I am getting excitation coming in from My sensory nerve fibers. Why are they exciting? That's the signal saying it's hot, right? And that means you want to initiate the withdrawal, so it's exciting that neuron. Does that make sense? You excite the neuron to make the withdrawal reflex happen. But what was your brain saying? Don't drop it. Don't, take your time. So those are actually inhibitory stimuli. You don't initiate those if it's just the reflex arc. The reflex arc is just in and back out without talking to the brain. But if you initiate it with brain input to begin with, you saw the boiling pot. So before you even grab the pot, you're already bracing yourself that it was going to be hot. Then you've got inhibition coming in on other dendrites, from other neurons in the brain. Okay? So what kind of integration are we going to have here? All you have to do is add up the voltages. Okay? So we open some excitatory synapses over here, and we're probably letting sodium in. Okay? And that's creating an excitatory potential. But on the synapses between this neuron and the brain, what are we doing? We're opening inhibitory channels, potassium or chloride channels. And so the effects can cancel each other. So if I'm letting in sodium over here, but over here I have potassium gates open, potassium's going out, they can just cancel each other out. And if they exactly cancel, then nothing happens. If the inhibition is stronger than the excitation, then you actually end up with hyperpolarization. If it's excited more than it's inhibited, but it's still subthreshold, then you'll have depolarization, but not threshold. So if one more stimulus came in, that might be enough to pop it over. So when you look at that on a graph, what does it look like? Our graph, remember, shows change in voltage over time. So if we're sitting here at rest, our resting value is -70. And then you saw the boiling pot, and at the same time you reached for it, what are you going to see here? Excitation, inhibition. If it was hotter than you thought it was, who's going to win? Whoop, you're going to get to threshold. And as soon as you get to threshold, what happens? Boom, you get an action potential, you initiate the withdrawal. Okay? So that's what integration means. That means pulling together all the excitatory impulses, all the inhibitory impulses, measuring the voltage at the axon hillock. And that's what this graph represents. It represents the millivolt change just at the axon hillock, because that's the only part that matters. Okay? So if we measured it at a dendrite, and this is just hypothetical because I don't think we can actually do that, but if this is a giant multipolar neuron, and here's my axon hillock at the bottom, if I could measure the voltage here, and that was the spot at which the synapses from my thermal receptors in the skin are hitting, what do you think that voltage is going to be? Is it going to go up or down? It goes up because you're exciting it. So you're going to get a little excitatory burst there. On the other hand, if I'm sitting at another synapse way over here, and here's where I have connections from my brain that say, I know it's going to be hot, but you're not supposed to drop it, then what do you think you're going to see over there? It's going to go down. So depending on which one is stronger, what matters is what's happening here at the axon hillock. And so if you're measuring it there, if it stays pretty much rock steady at -70, or you get a little bit of a blip, but it doesn't get to threshold, no action potential, no contraction. Okay, does everybody understand the basics of integration? Now multiply that by about 1 quadrillion, and you have what's happening in your brain. Okay, so when we try to translate that into what's happening in the brain, we put neurons together into circuits. And some circuits have more than one neuron in them, so we refer to them as pools of neurons. We have neuronal pools and neuronal circuits. And we have two different kinds of pathways, And then we have several different kinds of circuits we need to look at. First, let's look at our pathways. We have serial pathways versus parallel pathways. And serial means in a series. So if I have a series of neurons that interconnect, then I may have one neuron that stimulates another neuron that stimulates another neuron, and they're all going in the same destination. They're all part of the same function. That would be a series. That's pretty straightforward, I hope. Okay? I can make this parallel simply by setting up another line of neurons in the same pathway. Do you remember what we called a branch on an axon? That's a collateral. So do I do that? Is that a real thing? All the time, all the time. Okay, so do you remember we said that every motor unit is one motor neuron plus all the muscle fibers it innervates? Well, that's kind of, it's not really a parallel pathway, but it's an idea of spreading the message from one cell to multiple cells. Okay, so here's the cool thing about parallel pathways. What if these pathways were not sitting right next to each other, but they were actually, there was a split between them, and one went to one part of the spinal cord and brain and another went to another? Then if something happened, if I had trauma that killed this part, then it may not affect the other part of the path. So I may still have some of the function preserved. It may be diminished, but it's still there. Okay? So which one sounds better to you, serial or parallel? Parallel gives you redundancy, and redundancy means if you lose part of it, you might still retain some of the functionality. Memories are stored redundantly through parallel pathways. So is there some part of your brain you could excise to remove a single memory? Apparently not. They're diffusely stored through parallel pathways. Okay? Serial pathways, though, are found in some of the higher brain functions. For example, language functions tend to be just on the left side of the brain, not on the right, in most people. So you have a section of your brain over here on the left side that has unique functions that are not also preserved on the right side of the brain. The right side of the brain may have some impact, but it doesn't do the same thing. So if you have a stroke to a very small part of the left side of the brain, what you may find is that you have lost some of your linguistic abilities. You may lose the ability to name fruits and vegetables, that specific. So I saw an interview with a woman that they were demonstrating that, and they just set an apple on the table and they asked her what it was, and she said, "Oh, I love those." And they asked her to describe it, and she said, "Well, it's round and it's red, and you can peel it or not, and you can bake it into pies or you can eat it raw." And they kept saying, "What's it called? What's the name of it?" It's on the tip of my tongue. And she just flat couldn't remember it. So she knew everything about it, but she couldn't name it because of a stroke to a very tiny part of the left side of her brain. So sometimes it's that specific, sometimes it's in the wiring of grammar. So if I tell you the lion was eaten by the leopard and asked you who got eaten, what's the answer? Give me a minute. The lion was eaten by the leopard. Who got eaten? Everybody agree it's the lion. Okay. If you have a stroke to a part a little bit further back, you're sitting there going, I'm not really sure. I can't. Can you ask me that again? Can you reword that? You just can't. It's weird grammar. So if you say the leopard ate the lion, they get it. But as soon as you reverse it and use that weirder verb form, they lose it. So there are parts of the brain, particularly for language functions, that appear to have serial pathways. What does that mean? If it's serial, if you lose it, it's gone. There's no other part of the brain that can cover it. Okay? So, and we'll talk more about stuff like that when we get into the brain. But does anybody have any questions or concerns about a serial pathway and a parallel pathway? Okay, then we're going to look at our types of circuits. These circuit diagrams can be a little confusing. Let me see if I can make them just help you pick out the highlights of them. The names are going to help. If something is a convergent pathway, what do you suppose it means to be convergent? They all point to the same thing. So as you look at this picture, do you see the arrows all pointing to the same output neuron? They're all converging, okay? So when we talked about integration before, where we had the brain stimulating and the sensory neuron stimulating that poor little motor neuron in your withdrawal reflex, that was a convergent circuit. That was inputs coming from different places, all converging on the same output neuron. So what would the opposite of convergent be? That would be divergent. So here you see from a single input neuron that you have outputs going to different directions. And what kind of pathway is that? Look at that picture. What kind of pathway is that? That's a parallel pathway. Okay? So those are easy. Now let's get into the ones that are a little more confusing because they're harder to follow. Let's just follow one pathway. So from my input neuron, my original neuron, I'm going to go here, and then I'm going to go here. And then from there, I'm going to go here. And then I'm going to go here. And then I'm going to go here again. So where do I go from here? Back down. And around. And around and around and around. Can you see why we call it reverberating? It sort of echoes. Okay? And every time it does, we get an output. Every time it goes around, we get an output. Okay? So a reverberating circuit will last an indeterminate amount of time. Part of it depends on how many neurons are in the pathway, and part of it depends on things like your state of fatigue and things like that. But reverberating circuits, we think, are involved in processes that need to last for indeterminate but definite amount of time. We know that one impulse lasts about a millisecond. So what if I needed an impulse to go for 2 seconds? Instead of having multiple pathways to re-stimulate it, I can just set up a reverberating circuit that lasts about 2 seconds. And it will last that long because it will fatigue at the end of about 2 seconds. So when you inhale, what are you doing? How do you inhale? Y'all did muscles, right? What are the muscles of inhalation that you learned? Diaphragm and external intercostals. Okay, so imagine you have the phrenic nerves, which supply the diaphragm, you're learning that today, and the intercostal nerves, which supply the intercostal muscles. Okay, so you stimulate those for about 2 seconds, and so you inhale for about 2 seconds, and then that impulse stops, and so you exhale. So a reverberating circuit that lasts about 2 seconds is how we think the respiratory center in the medulla of your brain drives inspiration. And then it fatigues, and it takes about 3 seconds for it to recover. So if you're sitting there at rest, you're inhaling for 2 seconds, then you're exhaling for 3 seconds, and then you automatically inhale again. And we'll talk about how you do the automatic part of it separately. But basically, you have a reverberating circuit, we're pretty sure. Questions about that? Okay, the last one is a parallel after discharge circuit. Can you tell it's got parallel pathways in it? Okay, so let's count them. I have 1, 2, 3, 4. And notice, too, that each one of those pathways has different numbers of neurons. So this one has no neurons, just axons straight down. This one has 1 extra neuron. This one has 1, 2, 3 extra neurons. This one has 2. So because they all have different numbers of neurons, what difference does that make? Well, every time you hit a synapse, it takes a little longer for the message to get through. So we have a term for that. We just call it synaptic delay. So if you have no synapses, then this message goes pretty much straight through, zooms to the output neuron, it reaches threshold, it sends an impulse, and then immediately after that, then this one, let me try drawing that again. So this gets the impulse pretty much at the same time as this one, and this one. They all get the impulse at the same time. They all hit threshold. They all send an impulse. But this guy sends the impulse out. What does this guy do? Sends it to the next neuron. This guy sends it to the next neuron, which is the output neuron, so we get a second impulse. This one sends it to its next neuron, all those at the same time. And then we do that again, so we get this guy's ready, this guy's not, and then finally we get a fourth. So you get specifically and discretely 4 signals out with this setup. Not just sort of reverberating for 2 seconds, more or less, but exactly 4. Okay? So it's very discrete, this parallel after discharge circuit, and it lasts a very specific period of time. And I don't know of a single concrete example in the brain. We think that maybe parts of your short-term memory work this way. We think that they do, but we're not 100% sure. And there are things that can disrupt that. So we know from studies that have been done when telephones were first invented, but when telephone systems were first set up. How long is a telephone number? How many digits is a telephone number? These days it's 10. What was it originally? 7. So you have an area code, that's the extra 3. Okay? But if you know the area code, then that's long-term memory. But if you're just looking up a phone number long enough to dial, you got to call the plumber. You're going to look it up. Now these days, this doesn't even work. What do you do if you have to call somebody? You pull out your phone and you look, yeah, and you push the speed dial, right? Yeah, I do that even for my phone anymore. But if I had to look up a new number and I'm looking it up, guess how many digits most people can remember from the time they looked at the number to the time they can dial it on their rotary phone? That would be 7. Okay? And in fact, back in the olden days, we had the first 3 numbers, the prefix, was always a word, not a number. So, you have letters on your, analogous to your numbers. So I still remember the first phone number I ever had to learn. Do you? You remember? You had to learn one, didn't you? Didn't you have to learn your home phone number when you were a kid? You had to learn it for school, if nothing else. In first grade, they made you learn your phone number and the names of your parents. And we're paranoid these days. We quiz our kids on all that, teach them all that.
We learned it in kindergarten.
You learned it in kindergarten. I'm sure I did.
Have the same number.
You still have the same number, so that's kind of hard to.
Yeah, the same number for over 20 years.
Yeah, I still remember mine though. It started with Taylor. It didn't start with numbers. So, we have different memory tricks. But that's all important because if your number is too long, you can't remember the thing long enough to dial it. And what would happen if you had just looked up the number and you were just in the middle of dialing it when someone interrupted you and asked you a question? Boom, that's gone. Short-term memory, yeah, it doesn't work that way. So there are other, you have these memory pathways, and so you've got this, we think, parallel after discharge that lasts 7 numbers worth. But if something delays it, what happens? Those discharges are gone. So they're not there anymore. Okay, so that's the way we think that works. I have never read anything that says concretely that that's for sure the way it is, and I'm not sure how they would tell, but there are a lot of things I'm not sure how they figure things out. All right, but those are the those are the circuits that you need to know. You need to know the names, and you need to associate them with the diagrams. Okay, so what was synaptic delay? It has to go through multiple synapses. It slows the signal down. And which pathway was that important in? The parallel one, the after discharge. Could it be important in a reverberating? So if in my reverberating circuit, that's my output, and this guy's going to come back. If I stimulate it too quickly, then it may fatigue too quickly. So what would happen if, instead of that, I built in some extra synapses here? So what if I put in some nice little, just some extra synapses? And it's only this last one that's going to come back around and re-stimulate. Is that going to fatigue as quickly? No, because with the synaptic delay, it's going to slow down the rate at which each neuron is stimulated. So it may be a little slower, but it may last longer because there's less fatigue. Okay? All right, so now we're ready to focus our attention on the specifics. This is kind of molecular biology, neurophysiology combined of how the action potential does its thing. okay? And this is going to have clinical implications, so try to make sure you get what's happening. We know already that voltage gates are opened by voltage, which is electrical current, and that as you create electrical current, that opens the next voltage gates. So that's what propagation is. So in propagation, what we'll see is that we have voltage gates sitting at the axon hillock. What opened those gates? And where did that voltage come from? We're at the hillock, so where did that voltage come from? The dendrites. Remember, they had local action or local potentials, which created a threshold voltage. So as long as you get to threshold voltage, then these gates pop open, and as they pop open, that creates electrical current, and that opens the next gates. And then they pop open, and that creates electrical current that opens the next gates. And so it propagates all the way down the neuron. So what's missing from this picture? They don't show you the gates. So I don't want you to be left with the impression that this voltage change is occurring all on its own. You've got to have gates involved. All right, so we're going to look at the steps of a single action potential. Here's the picture that shows you your gates. And you got to remember you also have sodium-potassium pumps that are continually putting the, which way does the sodium go in a pump? And how many? 3 out and 2 potassiums in. Very good. So what we're going to see is the gates disrupt that, and then the pumps put it back to normal. Okay? So I'm going to show you the steps, and then I'm going to draw it for you because graphing is an important skill that we need to be working on. So we have the local potential, which is going to get us to threshold. And then once we hit threshold at the axon hillock, then the first voltage gates will open. And once you open the first voltage gates, then sodium is going to rush in. Do you remember I said sodium is more likely to rush in than potassium to rush out? So sodium in first. And because sodium's coming in, which way does the graph go, up or down? You're letting positives in, so it goes up. It depolarizes. And it's actually going to reverse. What does that mean? The resting potential was negative, negative 70. If I reverse that potential, it goes above 0. It hits a positive value, around positive 30. And that shift in polarity does 2 things. It causes the sodium gates to close, and it, at about the same time, fully opens the potassium gate. So which way does potassium want to go? It's okay to look at your notes from yesterday. Potassium wants to leave, and now it has 2 good reasons to want to leave. It used to be negative inside, and that kind of held the potassium, but we just made it positive inside. And at the same time, we closed the sodium gate, so the only way to make that positive leave is to kick the potassium out. So potassium leaves. We had it polarized, we depolarized it, now we're going to repolarize it. Potassium repolarizes. Okay? So sodium depolarizes, then potassium repolarizes. Okay? And we're going to have to talk about our sodium gates because they have some weird little features associated with them that we have to deal with. Okay, so there's the diagram. Maybe we'll come back to that in a minute. Here's the graph. So this should look similar to what I've been drawing. So you see you have time and you have millivolts. So you're looking at the change in the membrane potential. We're sitting here at negative 70 to begin with. We have our threshold marked with a dashed line. Are you with me on all those features? Okay, so look at the color changes when we go from a white background to a blue background. That is the onset of stimulation. Okay? We're sub-threshold. What's making us go up in this part right here? Voltage gates or local gates? Oh, you know what? I could turn those on. I'm so sorry. I didn't mean to leave you in the dark over there. I was having issues with the technical part, and then I forgot to turn these on. All right, so let's remind ourselves of what we were doing. We had a multipolar neuron, here is our axon hillock. So if I had stimulation out here, and it's excitatory, I'm opening sodium channels, then what happens is you get a local potential here. And how far can that local potential go? All the way to the axon hillock. And when it gets to the axon hillock, if we're graphing what the membrane potential at the axon hillock looks like, it was sitting at rest, minding its own business, until this voltage hit from the stimulation at that dendrite. And then what? Then it starts to go up, right? Okay, and then what? Then it hits threshold. And then what? What does threshold do? It opens voltage gates. And then what? And then sodium rushes in and takes us above zero. Okay, and then what? Sodium gates are going to close. Potassium gates have already probably started opening, but now they're going to fully open, and we're going to repolarize. Does everybody see how simple that is? Piece of cake, right? Okay, so looking again at the graph in your book. So what was our first step? That's our local potential from either synapses or stimulus gates or whatever. That's our local potential. You have to get to threshold before you start opening voltage gates. Okay? So once our voltage sodium gates open, then our potential goes, it just zooms up. How much time has passed from here to here? Just in that little lighter blue period there? Half a millisecond, that's probably, it's not even, it's more like a quarter of a millisecond. Okay? So that's the period of depolarization. So first we had a local potential till we hit threshold. Then we had a depolarizing potential, voltage-gated sodium channels. Then when we hit that positive 30, that does two things. What are the two things it does? Closes the sodium gates and opens the potassium gates. Okay? And then potassium rushes out. Potassium's positive, so our charge drops back down. Notice, please, that it drops below resting. That's called the period of hyperpolarization, and it's inevitable. It's just an artifact of the way it works. You have actually more potassium leaving than it took to counteract the sodium coming in. So you get briefly hyperpolarized. How much time till we get back to rest? About a millisecond and a half, more or less. Are you with me on all the steps? So can you name the three parts? If we call this the action potential, what are the three parts? The local potential is not part of the action potential. It precedes it. Depolarization, because the sodium gates open. Repolarization, because the potassium gates open. And then hyperpolarization, because more potassium left than it took to compensate for the sodium coming in. How do I get back to rest? How do I get back to rest? There's actually a couple things going on here. Number one, the ions simply diffuse away from the membrane, and they have a tendency just to correct things themselves. But don't forget you have those sodium-potassium pumps working. So what we found is you can stimulate one little neuron experimentally 1,000 times so fast that the sodium-potassium pumps can't compensate for it, 1,000 times before it runs out of gas, runs out of ion gradient. So you've got way more ions there than it takes to recover from an action potential. Okay? But at some point, the sodium-potassium pumps are chugging along, and they just keep throwing that sodium back out and pulling that potassium back in. So that's just kind of background. Okay? Questions at this point? Okay, let's focus on our sodium gates because they're unusual. Sodium gates actually are double gated. So if you look at this diagram, here is your sodium gate in its closed state. Here is a sodium gate in the activation state, and here is a sodium gate closed in its resting state. All right, so listen to the explanation and see if it makes sense. My sodium channels have two gates. only one of which can be opened by voltage. I have to have both gates open at the same time for sodium to flow. Does that make sense? If I close either one of the gates, the sodium stops flowing. But only one of the gates can be triggered to open. The other one kind of runs automatically. Okay, so if it's in its resting state, then I can trigger it to open. When it opens, both gates are open and sodium flows and we're in depolarization. But when we get to the peak of that action potential, the other gate closes. We call this the inactivation gate. And you can't do anything about that gate. You can't change anything to make that gate open or close. It has to reset on its own time frame. And what causes it to reset is a lower voltage. Okay, so let's take a look at our graph again, and we'll correlate with our graph the states of the sodium gate. So here, when we're at rest, here and here, we're in the resting state. So can I stimulate my sodium gates to open if they're in the resting state? Yeah, that's when I can. So when I hit threshold, that's when I open the activation gate. Okay? And what happens when I open the activation gate? Everything works, sodium's flowing. When I get to the top of the peak and my sodium gates close, it's the inactivation gate that closes. Can I reopen that gate? No. So I have to wait, and I'm going to wait until about here. Typically, I'm going to wait till about here. And then those inactivation gates will close, or excuse me, open, and the regular activation gate will close. Okay, and now I could re-stimulate it again. Okay, so let's list that, because I think that will help. Okay, so at rest, the activation gate is the one that's closed. We're talking about the sodium voltage channels. Okay, when I reach threshold, what happens? Activation gate opens and sodium flows. And that gets me until I get to positive 30 millivolts. Are you following me? Imagining this? Okay, then what? So at plus 30, the inactivation gate closes. And that makes the sodium quit flowing. Now, meanwhile, next door, what are the potassium gates doing? They're open and potassium's flowing, and it's the flow of potassium that brings the voltage back down. And when that voltage drops low enough, two things happen. The inactivation gate opens, and simultaneously, the activation gate closes. Okay, so here's my little handy high-tech demo. 2 gates, activation gate, inactivation gate, okay? So if I'm at rest, what are they going to look like? Activation closed, inactivation open. When I stimulate it to threshold, activation opens, channels open, sodium flows. As sodium's flowing, then when it hits plus 30, what happens? Inactivation closes. And then potassium running makes the voltage drop back down, and then what? Last step, boom. Both pretty much at the same time. One closes while the other one opens back up. Okay? So what this reminds me of is when you go to the petting zoo or the aviary at the zoo. They always have double doors. Why? keep the animals in the enclosure. So when you go to those places, particularly when you're going with little kids that like to barge ahead, what do you always tell them? Don't open that gate until I get this other one closed, right? Because one thing you don't want at the petting zoo is both gates open at the same time. Those little goats will be flowing. Okay? So always close the one before you open the other. All right? Can everybody picture those gates? This is important. It has clinical relevance because what it refers us to, the outcome of all this, is that we have what we call the refractory period. So here is a term that may be new to you. Refractory means resistant. And we use this in other contexts. For example, you may have a patient, a diabetic patient who skipped a dose, blood sugar got crazy, went into acidosis, is in a coma. That patient is now refractory to insulin. What does that mean? Have you ever heard that? Any of y'all ever heard that? Won't work. It's too late. You'll have to try to fix the acid-base imbalance before you try to give them insulin. I like to say my children, and now I can say my grandchildren, are refractory to my advice and, correction. So when I tell them what to do, they're like, don't hear it. Okay? So, yeah, in one ear out the other. So that's what refractory in general means, resistant. It seems weird to me, but if you remember that light refracts off of objects, what does that mean for light to refract? It either bounces off or it goes a different way or something. So that's kind of the same meaning. So when we talk about the refractory period of an axon, an action potential, what we mean is it's resistant to stimulation. You cannot re-stimulate it. Okay? So if right now I asked you to turn on these lights, what would you tell me? They're already on. You can't do it. So you're non-compliant with my command to turn on the lights, right? You can't do it. So when I tell you a neuron is refractory, what that means is, hold on a second, I'm already being stimulated. Don't tell me to stimulate again. You cannot tell me to stimulate when I'm already being stimulated. So just wait, okay? And it has to do with those gates. So look up here again. So let's see, inactivation gate, open, activation gate closed. Can I stimulate this guy? Inactivation is open. Activation is the one I can tell to open. So I can tell it to open, and now ions are flowing. All right, so we're going up, we're depolarizing. Can I stimulate it now? No, why not? It's already open. So once you've opened that gate, you have to wait. How long do you have to wait? This is closed. Can I stimulate it now? No, because that's the inactivation gate. It's not going to do anything I tell it. Now what do I have to do? What was the next step? After it's pretty much done repolarizing, it resets. Now can I stimulate it? Yes. Okay, so let's take a look at that. So if I'm in the depolarization phase, then what do my two gates look like? Activation, is open, right? Activation is open and inactivation is open. And ions are flowing. Can I re-stimulate it? Remember what stimulation means. It means open the gate. And the gate's already open. So it's refractory. If during this downward phase, if I look at it there, what do my gates look like there? Activation's still open, but inactivation is closed. Ions are not flowing. Can I re-stimulate it? No, because the activation gate is still sitting there in the open state. Now remember what activation means. It means open the activation gate. But if it's already open, I can't re-stimulate it. So during the period that the activation gates are open, you could hit it with a hammer and it wouldn't care. It's not going to open a gate. Gate's already open. So that's what we call the absolute refractory period. There is nothing you can do that will make that gate open because it's already open. Okay so far? All right. What's happening here? It's what we call the relative refractory period, which means you could re-stimulate it, but it's going to take a stronger than typically threshold stimulus. All right, so let's see what's going on here. Are my activation gates closed and inactivation open? Yes. Okay, so could I re-stimulate it? Yes, it's in the configuration that I can re-stimulate it, but look where I am. I'm farther than resting level, below threshold. So since I'm below rest, hyperpolarized, it takes a stronger than normal stimulus to get to threshold. You're just numerically having to add more voltage in. Does everybody see why that's the relative refractory period? Can everybody pick out and explain the absolute refractory period and the relative refractory period? Okay, we're going to see that the same concept of refractory period applies to muscle as well. With skeletal muscle, it recovers before contraction, so we're not worried about that. But with cardiac muscle, it's going to be fundamentally very different. And in cardiac muscle, it's still refractory while it's contracting and relaxing. And it's important that it be that way, okay? What it prevents is tetany. What was tetany? How did we get to tetany? You had to re-stimulate it, and you stimulate it while it's still contracting, right? And it just stays contracted. Do you want your heart to do that? No, I want my heart to pump, which means it has to alternately contract and eject blood and relax so it can fill with new blood. Make sense? So pumps always have to alternate contracting and relaxing. So I don't want tetany in my heart. And I'll explain in AMP2 how we manage to do that. But it has to do with refractory period, okay? When you are fatigued, that can impact not only how much ATP you have available, but it can actually change the threshold. So here's the effect of fatigue on the threshold. What did I just do? You see I made a bigger distance between rest and threshold? So when you're so sound asleep, you sleep through your alarm because you're exhausted, you're getting normal threshold stimuli and the sound coming from your alarm clock, but your thresholds are elevated so that it's harder for you to respond to them. So guess what caffeine does? Caffeine lowers the threshold. It makes it easier for stimuli to get you to threshold. Okay? Sedatives like barbiturates raise the threshold. Okay? So does everybody understand the impact of threshold on this? All right, well, that's pretty much the ins and outs of a single action potential. But what we have to do is we have to get from a single action potential to a propagation. So what I'm going to find is that I'm going to start my action potential here at the axon hillock. So I get an action potential right there at the axon hillock, and that creates some electrical current, right? I have sodium and potassium ions flowing. And that electrical current can flow, just far enough. So it's going to flow just to the next set of gates. So when the next set of gates reaches threshold, what do? So we're to here now. It's going to have its own action potential. And when I get to the next set of gates, it's going to have its own action potential. So every little spot where there are gates has one action potential after another, all the way down the axon. What caused that action potential to propagate down the axon? The secret is these are all voltage gates, all the way. And what opens voltage gates? And how did I create voltage? By opening voltage gates. So it propagates because it is electrical current that is both the cause and the result of opening those gates. Does everybody understand propagation?
And you say it's because the ions are flowing.
So when you open those sodium voltage gates, sodium ions are just flooding into that cell, and that creates the electrical current that not only raises it to plus 30 there at that spot, but also travels through the axoplasm to the next little spot. Okay? All right. There are two different arrangements of axons. And what we're seeing here at the bottom is a myelinated axon. And myelin provides electrical insulation. So if that one's myelinated, what do you want to call this guy at the top? This one's called unmyelinated. It makes a difference, and here's why. If I look at the voltage gates for sodium and potassium, plus don't forget I need those sodium potassium pumps to put everything back, where I find them on my axon is anywhere that it's not covered with myelin. So how many gates do I need on an unmyelinated axon? They are going to be found all the way down the entire length. Okay? What about for my myelinated guy? Only at the bare spots. Which one looks like it has more gates to you? Unmyelinated. Which one seems more efficient to you? Myelinated. Which one seems faster to you? Myelinated. And the reason it's faster has to do with a couple of things, one of which you can tell from this picture and one of which you can't. So imagine that it doesn't matter how far apart the gates are, the time is spent on opening the gates and generating an interaction potential. So the more distance I can put between those gates, the fewer gates are involved, the more efficient it is because I don't have as many gates, but also the longer I can traverse in one time. Okay? So imagine taking a long stride and getting across this room versus taking baby steps. Which one's going to take longer? Little baby steps. Okay? So think of your unmyelinated, we call that continuous propagation, as being like baby steps. And so it's a little bit slower. But there's another factor at play here, and that has to do with how big the neurons actually are. So if it's unmyelinated, if we look at a cross-section of an unmyelinated axon, this is, I'm going to try to draw it to scale. It's not going to be great, but it'll help. I have 3 sizes of axons. I have some pretty large ones. We call these A, these are B, and these are C. So we have 3 sizes of nerve fibers that we just designate A, B, and C, and A is the biggest one. A and B are both myelinated. Why is A the fastest? Remember that we're talking about myelination, so we're talking about just at one little spot, just at that spot, that's where the gates are. So if I'm looking at one of these, we call them the nodes. If I'm looking at a node on A, an A fiber, and a node on a B fiber, what's the difference? A has more. Why does A have more? It's bigger. More surface area. Larger diameter means more surface area. More surface area means I got more room to put gates in. The more gates I have, the faster the ions can get me to threshold. Okay? So if I time these, if I look at an A fiber with its myelin sheath, and a B fiber with its myelin sheath, and a C fiber without a myelin sheath, and I stimulated them all at the same time, and I had them all go the same distance. Let's say they all had to go 1 meter. Then how long would it take for that impulse to go 1 meter along those 3 fibers? And what we find is that for our slow guys, our C fibers, it takes 1 to 2 seconds. And for our B fibers, it takes about 1/10 of a second. And for our A fibers, it takes about 1/100th of a second. In other words, an A fiber can carry an impulse 100 meters per second. A B fiber can carry an impulse 10 meters per second, and a C fiber can only carry it 1 or 2 meters per second. Does everybody see the difference? Now, how does that translate into reality? Your big fibers to your skeletal muscles are fast. So how long does it take an impulse to travel from your spinal cord to your foot? That's about a meter, about 1/100th of a second, okay? So if we also have fast sensory fibers, and when you step on a sharp object and you do that withdrawal reflex, how fast are those impulses traveling? A fibers all the way. It's faster than you're really conscious of, right? You're talking about very, very short time, half a second. Okay? Make sense? Now, I'm not going to ask you to do this on purpose, but I want you to think back or think forward the next time this happens to you. When you step on a sharp object, does it hurt right away? Stub your toe. Does it hurt right away? Usually you have just enough time to think, oh, expletive deleted. That's going to hurt. Just any second now, that's going to hurt. And then that wave of pain hits. You have just enough time to realize you did something dumb, and then it hurts. Okay? So guess what kind of fibers that dull, aching pain is traveling on? That would be C fibers. They're slow ones. So you get the A impulse for your withdrawal reflex because you need to pull back fast. And then the C fibers carrying the pain message comes in right after that saying that was a dumb thing to do, don't do it again. So touch a hot stove. You withdraw and then it hurts. Step on a sharp object, same thing. Okay? So what's the purpose of the pain? Does the pain induce the withdrawal? The pain is so you'll learn not to do that again. Okay? So it can come in immediately after, so you'll associate cause and effect, but also you'll already have withdrawn out of trouble. Make sense to everybody? Okay, we also find B and C fibers are going to control our visceral functions, like regulating the heart, regulating the digestive tract, kidneys, any internal organs that have a nerve supply. We use B and C fibers there, and so we'll study those on Thursday. Okay? So A fibers, where are they? To and from the things you're conscious of. You have voluntary control of or conscious awareness of. So your conscious sensations, A fibers. Okay, other than pain. Pain special. Okay, so we got off on that because we were talking about propagation and looking at the difference between myelinated and unmyelinated. We use the term saltatory to refer to myelinated propagation. Saltatory comes from the Latin saltare, which means to leap or to dance. So saltation is what we call it when you leap over the myelin parts and onto the nodes. All right, so that brings us to the problem of what happens if you lose your myelin sheath. This occurs in demyelinating disorders like multiple sclerosis. MS is an autoimmune disorder. We know a lot more about it than we used to. I haven't kept up. I don't mind telling you that. Because it affects just the myelinated nerves, what happens is that they lose their function. So let's see why that would be. So if I have a myelinated axon, where are my voltage gates? They're here and here and here, right? We call those the nodes. So you have gates only at the nodes. Okay so far? All right, what happens if you lose the myelin? Where are my gates? My gates are still sitting where the nodes used to be, but I've lost the myelin in between. So what happens to my electrical impulse? It fizzles out before it gets. Remember what the myelin does? It insulates, electrical insulation. So instead, remember that the flow of ions is decremental. It decreases over space, over distance. So it dies out before it gets there. It just never gets to the next node without that layer of myelin. Okay? This can happen slowly over time. Generally, it happens in episodes. So something, we don't know what, will trigger another round of demyelination. Could be sensory or motor. right? So you've got your big sensory nerves bringing sensations from the body, and then you have the motor nerves to the voluntary muscles going out. So you can lose both sensory and motor functions with MS. And so you wind up with weakness in the skeletal muscles, wind up in a wheelchair, but you also lose sensory function. And a lot of times that's the first thing, and people just don't notice. Or they think it's something else, okay? Over time, what might happen is that those nodes will kind of spread out. So they realize, in a way, that there's not a myelin sheath there. And generally what I think is probably really happening is that the cells are producing new channels, they're recycling their membrane, and since there's no myelin up there, they just part more gates. So the gates get redistributed over time, and you may regain the function, but is it going to be as fast as it was? Without the myelin, no, it's not going to be as fast. Okay, so what you find generally is people with MS will have a crisis, they'll lose some function, and then they'll regain some. And then they'll have another crisis and lose more function, and then regain some. So that's the only explanation I have based on neurophysiology. Corey?
I know that they have some type of like stem cell type surgery or something that people have been able to like walk again. What exactly is happening with that?
I have no idea. I didn't know they were doing stem cell for MS specifically. I didn't know that.
Yeah, I heard about that too.
What did you hear? What do you remember? Anything?
No, not really.
Nothing in specific? So Google that and
Yeah.
see what you find out. Yeah, I honestly don't keep up with. I read stuff. If it pops up in my news feed, I read about it, but I don't generally go searching for it. So usually my students are the ones that tell me about new things. The only research I know of that's been done on MS is to try to figure out what's causing the demyelination. We think macrophages are attacking the myelin sheath. We're not really sure what triggered it. But macrophages are hard to go after. If it were antibodies, you know, we know how to clear antibodies from the blood. If it's T cells, we know how to inhibit T cells. But macrophages, I don't know of a way to inhibit macrophages. So if the macrophages are doing the main attack, then I'm at a loss to explain it. If it's antibodies that are tagging the myelin sheath and then the macrophages go after it, then maybe we can do something with antibody therapy. But I don't know what. So it's one of the more intransigent conditions, just hard to get a handle on it to treat it. And stem cells, I don't even know where they would go with that. All right, so any questions about axons and what they're doing? Because we're going to move into new territory here. Okay, we've got to talk about our neurotransmitters. And this gets a little tedious, let me say. Okay? So what I'm going to suggest you do is use flashcards to keep them straight. You've got a list of neurotransmitters you need to learn. And so you can imagine in the test bank, I have at least one question on each one. And what are your multiple choices going to be? Since on my test, I'm not limited to 4 or 5 multiple choices, I just put the whole list. Yeah, so you got to be able to tell them apart from each other. Okay? So make sure you can do that. All right? Okay, so we have different categories of information, and we use these. So you need to learn them. You're going to hear of monoamines. You're going to hear of monoamine oxidase inhibitors. So you have to know the chemistry of the neurotransmitters in order to understand the drug classes involved. So our chemical classification we have looks like 1, 2, 3, 4, 5 different classes, and the 5th class is kind of a cheat. Everybody else is in that class, miscellaneous category. Acetylcholine is in a class by itself. Chemically, acetylcholine has 2 parts. Anybody want to guess what the 2 parts are? This is a trick question. You have an acetyl part and a choline part. And choline is a fairly complex organic molecule. Acetyl is just 2 little carbons with some hydrogens thrown in a little acid group. Acetic acid is vinegar. Okay, so it's the same thing. Acetyl, acetic, it's basically the same chemical group. Very cheap, very throwaway, very replaceable. So acetylcholine is made of these 2 parts. The acetyl group gets added to the choline to make it active. So to inactivate it, we know the enzyme that inactivates it, right? Acetylcholinesterase. All it does is break the acetyl group off and throw it away. And then we're going to see that we keep the choline, we recycle it. 3 different amino acids. Glutamate is one of the 20 amino acids. Glycine is one of the 20 amino acids. GABA is not. GABA stands for gamma, that's a Greek letter, gamma amino butyric acid. And I'm just trying to impress you, because I'm never going to ask you to remember that. Just call it GABA. Okay, what I did, and this is probably on another slide, but it helps to have it right here. Glutamate excites, as I explained yesterday. GABA and glycine inhibit. So just because they're all made of amino acids doesn't mean they have common elements. They're different amino acids. They have different pathways. They have different drugs that are going to act on them. But when you get to the monoamines, particularly the catecholamines, you're going to see that word. And so you need to know which ones are catecholamines. Now, we're not going to get bogged down in this. I'm not going to ask you to look at the chemical structure. I know the book gives you the chemical structure of these. I'm not going to ask you to recognize them by looking at their chemistry. Does anybody feel cheated by that? Okay. Serotonin is also known as 5-HT. And there are at least 5 different kinds of receptors that I know of. Serotonin is one of my favorites because it does such great things for you. It calms you down, it suppresses your appetite, it perks you up, makes you alert. What's not to like about serotonin, honestly? Okay, it also causes hallucinations and, you know, whatever. Okay, neuropeptides. What's a peptide? It's a little short chain of amino acids. So not a single amino acid, but a little group of amino acids. Okay? So you just need to know that they're in that class. And then others. What we're finding is that there are some gases that have neurotransmitter effects. That just blew everybody out of the water. We just flat didn't know that until it was discovered. Now, when physiologists discovered the role of nitrous oxide in triggering changes in smooth muscle behavior and blah, 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 all the botanists are going, old news, we knew that. If you want your fruit to ripen early, what do you do with it? If you want it to ripen, do you know what to do with fruit to make it ripen? If you want it to be soft but not mushy, put it in a paper bag with other fruit. Because ripe fruit gives off ethylene gas. It's a gas. And what they do commercially is they pick the fruit unripe, of course. Because if they waited till it was ripe before they picked it, what would it look like by the time it made it to the grocery store? So they pick it unripe, and then what do? They put it in a storage warehouse, and they pipe in ethylene gas. And that at least makes it look ripe, even if it doesn't taste ripe. Okay, so botanists have known for a long time that gases have effects, can trigger things, but we didn't. Okay, so that's one class, the chemical classification. Our second classifications, where are they found? Are they found just in the central nervous system? Are they found in both central and peripheral? And in fact, the list of neurotransmitters that acts at peripheral nerve endings is very small. So I want you to think about that. Based on what we know about reflex arcs, here's my sensory neuron. It comes into the brain or spinal cord. It synapses with my association neuron, which synapses with my motor neuron, which goes out to a muscle or a gland. Where are my synapses? I have synapses here and here and here, right? So which are the only synapses in the PNS? On the muscle, or if it's not a muscle, it's a gland. Okay? And if it's a muscle, if it's skeletal muscle, we already know that neurotransmitter. What is it? Acetylcholine. If it's a gland, or if it's skeletal muscle or cardiac muscle, it might be any one of these things. And we'll worry more about that on Thursday. So before you take your test, you'll have this down. But if it's not on this list of things found in the PNS, That's a short list, right? Then it must be in the CNS. Now I'm going to tell you, I should have been a little more specific. There's a whole set of neuronal pathways in your gut. We call it the enteric nervous system, and it has its own little set of rules. But in general, it's still going to be acetylcholine, epinephrine, norepinephrine, and potentially dopamine. Okay. Do you find this list of PNS guys? Are they found in the CNS? Yes. So they're found in both CNS and PNS. Okay? So it's either CNS only or it's both. And if it's both, what's our list? Acetylcholine, epi, norepi, dopamine. Are you with me so far on location? Okay, action, either excitatory or inhibitory, or it depends. And an example of it depending, if you put acetylcholine on skeletal muscle, what does it do? It contracts, right? If you put acetylcholine on your heart, it slows down. Okay, so same neurotransmitter, exactly the same stuff, but it can be excitatory in some places and inhibitory in others. Okay, same with norepinephrine. Okay, so you have in your online materials, I can't right now remember where, either in the chapter summary, the practice quiz, or both, you have a list of neurotransmitters. And I hope, if you don't, I'm going to make sure you do. I haven't looked at that chapter summary in a while. But you should be aware of it. Now, some of these things are going to be found in specific parts of the brain. So when we get to studying parts of the brain, which is, tomorrow, then we'll look at some specific examples of neurotransmitters and where they have their effects. And we'll also talk about some of the disorders associated with problems in the brain, and some of those are neurotransmitter specific. But some of the drugs that we use are associated with the neurotransmitters action. So I told you serotonin was my favorite. Have you ever heard of SSRI? What is it? Selective serotonin reuptake inhibitor. So what does that mean? So it selectively prevents serotonin from being taken up in some places, but not in others. Bipolar, ADHD, other disorders, it depends. Okay, so I have a special project I want to work on, and we worked on this a little bit before. Didn't we talk about Sevendust and Rotenone? Yeah, okay. So we're going to talk about that from the standpoint of drugs that interact with synapses, because that's where neurotransmitters do their thing, okay? And on your test, you're going to have some specific clinical problems associated with synaptic function, all right? So let's start with what happens at a synaptic bulb. And see under normal conditions, here's a synapse. All right, so let's label our cells. Which one of these cells is the presynaptic neuron? The blue one or the purple one? This is presynaptic. So it's going to release the neurotransmitter, and then that makes the purple one what? Postsynaptic neuron. So it's going to have the receptors. So when I stimulate this neuron adequately, and I do that through electrical voltage, hits the calcium channels open, calcium enters the cell. Y'all remember all that? So calcium comes in and triggers exocytosis, and I get exocytosis of my little neurotransmitter, and my neurotransmitter crosses the cleft and binds to the receptor, and then the receptors open the channels, right? Okay, so we'll, for the sake of simplicity, assume this is an excitatory guy and it's going to start a nerve impulse. Okay, so what else do I need? I need to be able to turn that signal off, don't I? So I'm going to have either in the synaptic cleft or maybe bound to the membrane, I'm going to have an enzyme that is going to break down my neurotransmitter. Typically, that's what I'm going to have. And I need one more piece that I haven't introduced to you yet. In the presynaptic membrane, I'm going to have reuptake pumps. These are active transporters. They're going to take the neurotransmitter back into the cell that secreted it, repackage it into new synaptic vesicles, and recycle it again. Are you with me on the big picture? Let's go through one and see. So what we already know is, in the case of, say, acetylcholine, we know that once the stimulus hits the synaptic knob, acetylcholine is released in the cleft, it diffuses to its receptors, it binds, it opens, channels open, right? Okay, and then I have my enzyme, acetylcholinesterase. What does it do? It breaks the acetyl group off of the choline group. And once they're separated, it can't bind to the receptor anymore. But what I haven't told you till right now is that there are choline reuptake pumps. We have choline reuptake pumps. Choline's expensive. We're not going to waste this stuff. We're going to throw away the acetyl group. It's just going to diffuse out of the synaptic cleft. It's going to get picked up by neighboring neuroglial cells or macrophages or diffuse into the blood or whatever. We don't really care what happens to the acetyl group. It's just acetic acid. We throw it away. But choline's expensive, so we recycle it. And in that synaptic bulb, I've got all I need to be able to rebuild it into acetylcholine, repackage it into a new synaptic vesicle, and use it over again. Are you with me on that big picture? So I had two steps, though, right? First, I deactivated it, and then I pumped it, right? And that's faster deactivation than if I just pumped. Okay? Because pumps are slow. But there are some things that are only pumped. They are not deactivated. They are just pumped. All right? An example of that is dopamine. So dopamine is like my second favorite after serotonin. I like dopamine. It puts me in a good mood like serotonin does. It also gives me control of my muscles. So dopamine deficiency is associated with Parkinson's disease. So people with Parkinson's are not usually in a good mood. You wouldn't be either if you had Parkinson's, right? So for years we just dismissed it. Of course he's not happy. He has Parkinson's disease. Yeah, it actually is a physiological effect. Okay? Dopamine doesn't have an enzyme to break it down. All dopamine has is a reuptake pump. Are you with me on that? So if my reuptake pumps are not very efficient, what happens to the dopamine? It'll sit there longer, and that will accentuate the effect of the dopamine, but eventually it will diffuse out of the cleft. And what happens to things that diffuse out of the cleft? They're gone and not recycled. Does everybody see the setup? Okay. Cocaine blocks the reuptake of dopamine. Cocaine is a dopamine reuptake pump blocker. So tell me what that means. What did dopamine do? Makes you happy. It really does. Makes you feel really good. So what does cocaine do? It keeps the dopamine in the synapse longer by blocking the reuptake. So you feel great. And I'm just for the record, not from personal experience. Okay, but then what? What happens when the cocaine wears off? Then you crash. Why? What happened to all that dopamine? You were like throwing money away right and left, and then you were impoverished. You had nothing left. Okay, so this is like taking your whole paycheck on Friday, going out Friday night, having a great time, and then being hungover and broke the rest of the weekend. Because what are your cells used to? What are they programmed for? Recycle the dopamine. Reuse it. And now you're like, I don't have any for you to recycle and all that. So it takes them a while to reaccumulate some dopamine because that's not the way they're programmed. Okay? So when you crash, as the cocaine wears off and the dopamine's depleted, then you don't feel normal. You feel horrible because you got no dopamine. And it takes a while to recover from that. And honestly, some people never get over that sensation of feeling so great. They just, one-time use of cocaine just does them in. They're never the same. Now, there's another condition that involves this dopamine. And that one you may have heard of. It's called Tourette's. Have you heard of Tourette's? What do you know about Tourette's? Okay, so we call them tics usually. Anything else specifically anybody knows about Tourette's? Let's hear it. Come on. Speak up. Nothing? They have outbursts and they have weird tics. They'll copy people. They'll mimic people. They'll dance down the sidewalk. They'll ape one person and start following another and just go back and forth. They may have facial tics. What else? Emotional outbursts. They have verbal tics. So what's a verbal tic? A facial tic is when that muscle keeps contracting over and over. What's a verbal tic? It's not a stutter usually. They're usually quite eloquent.
Say something out of nowhere?
Not just out of nowhere. They curse. They curse, you guys. So I'm going to tell you, this is something that really relates more to tomorrow, but we may run out of time tomorrow. They're the parts of your brain that are involved in language. You have 3 language circuits in your brain, 2 of which are easily distinguishable in nonverbal animals. So birds have alarm calls, for example, and they may have a different alarm call for a flying predator than they do a snake on the ground. And if you study them, you learn the differences. Primates particularly will have a different alarm call for different situations. So you have alarm calls. You also have gestures, right? We have a very rich gestural language. Yeah, so do other animals. And then we have what we call our higher language functions, our grammatical-based cognitive functions of language, which are kind of laid, the wiring of that is laid on top of those more primitive ones. So alarm calling definitely connected to emotions, right? And gestural language, is that connected to emotion or what? What usually triggers gestures? Yeah, it's usually, yeah. Okay, so when someone has Tourette's, preferentially it hits those primitive circuits, which in humans are expressed as obscene language, cursing. Don't know why, but it probably has to do with that emotional aspect of it. And I can't really say I've seen them gesture, but they do have, different motor outputs as well. But it's legendary, Tourette's. You just associate that with cussing. And you could have a 2-year-old with Tourette's, and they're going to say whatever they learned as cuss words. What does a 2-year-old know as a cuss word? A 2 or a 3 or a 4-year-old. It's words that it would make you laugh. They're so cute. Except to them and their vocabulary improves as they get older. Okay, so what's up with Tourette's? Tourette's is actually an excess of dopamine. Whereas Parkinson's is a deficiency of dopamine, Tourette's is an excess of dopamine. And so it expresses that excess in terms of locomotion, because dopamine is for motor control, but also that emotional aspect. How do people with Tourette's feel? How do people with cocaine feel? How do people with Parkinson's feel? They're just like blah. They have no good feelings because they have a deficiency of dopamine. So people with Tourette's have an excess of dopamine. How do they feel? They feel great. So if you have a kid, teenager, whatever, a young adult with Tourette's, you're sitting there saying, honey, you've got to get a job. You know, you've got to find somewhere to work. You're worried about their independent living, you know, for the rest of their lives. And how do they feel about that? They don't care because they feel great. Okay, so the therapy for someone with Tourette's is very difficult because in their mind, they don't have a problem. They feel fine. So once they're on their own and independent, young adulthood usually is when that hits. Some of them maybe a little earlier than that. Then they start getting this feedback of, gee, I'm hungry and I have nowhere to live. I guess I ought to go get a job. Well, how good are they at holding jobs? Well, if they have these outbursts, that's not good, right? So luckily there's a treatment. There's a drug that suppresses dopamine production. So you just put them on this prescription drug, and it suppresses their dopamine, and they're not cured. It's a lifelong treatment, as far as I know. So if you start it when they're little kids, if you get them diagnosed early and get the dosage just right, then they never get hooked on that great feeling that they have. They just control it lifelong. But if they're older, when they're diagnosed, and you put them on this drug, what's one of the side effects to them? A side effect of this drug. They don't feel good anymore. You've lost that great feeling that people use cocaine till they're dead to get. I mean, you know, seriously, it's that strong of a compulsion. So you put them on this drug so they can control the unpleasant side effects or the unpleasant effects, and to them, and to some of them, it's unbearable. They will not give it up. Others find that because the half-life of this drug is pretty short, wears off about the same time frame as caffeine. So if you take your dose in the morning, you suppress the urges to have these outbursts while you're at work so you don't get fired. And then when you go home, you can feel as good as you want because you're off the clock. So a lot of people who have Tourette's, well, of the few cases that I've read of, that's been the most successful in terms of managing it. So they can feel themselves, feel normal only when they are off this drug, and this drug literally masks any good feelings that they have. So it's not, to me, a great treatment. It has a lot of recidivism. You know, people just go off the treatment because it makes them feel so good. So best is to start them when they're little kids so that they never get hooked on that high that you get from the excess of dopamine. All right, so as we go through the class tomorrow and Thursday, we'll be coming back to our neurotransmitters and applying more of them. But we have looked at several different aspects of the synapse. We already looked at things that can block exocytosis. We've looked at things that can block the receptors. We've looked at things that can block the enzymes that break it down, and now we've looked at things that can block the reuptake. Okay? So in your own study, you need to know for each one of these cases, does it inhibit or enhance the effect of the neurotransmitter? Okay? All right, finally today, we need to talk about the neuroglia. And I know I've introduced the neuroglia, and you have a nice picture in your book. It's very pretty. But we need to look at what are the characteristics of each of the kind of the neuroglia and what, if any, clinical problems are associated with them. And what you're going to find is that the first distinction we make is between the glial cells of the central nervous system and the glial cells of the peripheral nervous system. Okay? So there are going to be similarities, there are going to be differences, and you need to be aware of them. Additionally, you're going to find slight differences in the brain compared to the spinal cord. Even though the glial cells are found in both, there are going to be some differences. Okay, so let's start a list of our glial cells. So we had our main distinction was CNS and PNS. And in the CNS, we have 4 basic kinds. We have astrocytes. Tell me what their shape is. Star-shaped. We have oligodendrocytes. What was dendro? Branches. And what was oligo? Few. So these are cells that have just a few branches. We have ependymal cells. And we have microglia, or microglial cells. Ependyma means coat. So the ependymal cells are going to coat, that is, line the cavities of the brain and spinal cord. Tell me, of these 4 kinds of cells, which one do you think is the smallest? That would be the microglia. Okay. In the PNS, we only have 2 kinds of cells. One of the cells kind of hangs around the neurons, so we call it a satellite cell. And we really don't know much about them, so I don't really have a lot to tell you about that. The other cells we still usually call after the person who first described them, even though we're trying to get away from names named after people. The Schwann cell. And the reason is the other name of the Schwann cell is just cumbersome. Neurolimocyte. Which would you rather call it? Schwann's just easier. So 4 in the CNS, 2 in the PNS, and really literally no mixing between the 2. Okay? So those that have been in the lab this morning are in better shape than those that haven't, because you can answer questions that the afternoon lab guys aren't ready to answer yet. If this is the spinal cord, and here is the posterior root and the posterior ganglion. And here is the ventral root. And there's the spinal nerve. Is this structure PNS or CNS? PNS. And what about the spinal cord, of course? That's CNS. So likewise, if I'm looking at the brain stem, which is just a little lumpy part on the base of the brain, the ventral side, I find some nerves coming off of it. And if along those nerves I find some little swellings, those are called ganglia, by the way. Are those PNS or CNS? Nerves are PNS. The ganglia, the swellings on the nerves, are PNS. Okay? Brain and spinal cord are CNS, and that's all there is. So if we go back to our list, where do we find astrocytes, oligodendrocytes, ependymal cells, and microglial cells? Brain and spinal cord. And where do we find satellite cells and Schwann cells? Satellite cells are in the ganglia. Schwann cells are in the nerves. Okay? Satellite cells are in the ganglia. Schwann cells are in the nerves. And I'm going to show you that real quick. So here is that posterior or dorsal root again. And here it is coming into the spinal cord. That is the ganglion. Ganglion just means knot, like a knot on a thread. And if I drew the part of the reflex arc that y'all are learning today, then we would see that sensory axon coming in here. That's where the unipolar neuron cell body is. And then it's going to get into the spinal cord to make its synapse. So where are my satellite cells? They're around the neuron cell body. Okay? And where are the Schwann cells? They're here. They're associated only with the axon. Are you with me on that? Okay. Let's go back to the big picture in the book. And here what I want to point out is that there's myelin in both. The Schwann cells give me the myelin sheath in the PNS. We call it the neurolemma. But I also have myelinated axons in the CNS. I have myelin in my brain and spinal cord. And as far as the axon's concerned, it works the same way. You get that nice saltatory conduction from one node to the next node, whether you're in the CNS or the PNS. But you have totally different neuroglial cells that give us that product. So in the CNS, it's the oligodendrocytes that give us the myelin sheath, whereas in the PNS, it's the Schwann cells. And you're going to have, if you have MS, it could affect either one of them as far as I know. Okay, so who does myelination in the CNS? CNS, oligodendrocytes. PNS, Schwann cells. That's a distinction I expect you to remember. Okay, so far? One of the most important kinds of neuroglial cells is the astrocyte. It's this big green thing. Can you see the big green thing? And it actually has what to me look like little duck feet. And those little duck feet actually reach out and grab the capillary, and another little duck foot will go over, reach out and grab the neuron. Now, what's it doing? For a long time, we thought it was just structural. It was actually holding the neurons to the blood supply. And if you have a hemorrhagic stroke, what does that mean? A hemorrhagic stroke means you're bleeding. You've broken a blood vessel and you have blood coming out, it's gushing out. Then you rip these connections between the astrocytes, the neurons, and the blood supply, and that adds to the damage of the loss of blood. If you have a thrombolytic stroke, then you have a clot that's stopping the flow of blood, so you don't have that much damage. You just have a loss of oxygen and nutrients. But a hemorrhagic stroke, boy, you've got some trauma, some shearing force in there. So we blame the astrocytes for that. And for a long time we thought, well, that's what they're doing. Well, you know what? They're doing a lot more than that. So astrocytes are absolutely crucial. They're the most important kind for you to learn. You need to learn what they do. They are regulating the extracellular fluid. That means they're controlling the electrolyte balance, the sodium and potassium ions, the pH. They also, it turns out, are feeding the neurons. And this was, to me, mind-boggling. Astrocytes can take glycogen the same way muscles do and break it down to lactic acid. And then they can feed that lactic acid to neurons, like baby food. Feed my babies. Astrocytes can feed the neurons. Who knew? We also now know that astrocytes actually talk to each other through gap junctions. Remember what gap junctions are? They're the little tunnels between cells that are physically in contact with each other. Neurons have nerve impulses, voltage changes due to sodium, potassium voltage gates. Astrocytes have gap junctions, calcium's doing the talking. But when a neuron sends an impulse, these astrocytes get excited and they start sending these calcium signals, and honestly, we don't really know what they're saying. We just recently found out they were talking to each other. Finally, astrocytes form scar tissue in the CNS. Now, this is good and bad. You need scar tissue, why? Doesn't beat having a gaping hole. Okay? So everything else being equal, that's great. But if you lost some connections, if axons got cut, then as the astrocytes form scar tissue, they block the regrowth of axons. So picture this, the neuron cell body's intact, but the axon got traumatized, sliced in two. The neuron cell body will try to grow a new axon. What stops it? Those troublesome astrocytes have gone and put some scar tissue in the way. And we know this is true, and particularly in spinal cord trauma. It becomes a goal to try to find a way to circumvent that scar tissue formation and let those axons regrow. Now, when I started teaching in the 1980s, at the time I was reading up on this, and they said, in 10 years, we're going to have this. Yeah. I'm still going to tell you it's about 10 years out, which is our way of saying we don't know when. So we are further ahead in applying exoskeleton technology and electrical stimulation of muscles than we are in fixing a broken spinal cord. So we'll get a physical and engineered solution before we have a biological solution. There's some early research that shows if you put a graft from a peripheral nerve into the spinal cord, where you have Schwann cells instead, that it can guide the regrowth. And we know that works in the peripheral nervous system. Have you ever cut a nerve? Anybody had an injury deep enough to cut a nerve? Well, those axons do grow back. They grow back at the rate of about 1 centimeter a year, not real fast. So if you had an injury in the elbow, how long before you might get some feeling back in your fingers? 1 centimeter a year. Okay, my husband actually had trauma to his thumb. It's a long story, but don't let two male iguanas in the same room. They tend to fight. And then don't get between them if you do. So big gash. And he lost sensory function in the thumb. And in a couple of years or so, he said, oh, I can feel my thumb again. So the axons will regenerate. The Schwann cells will guide the regeneration. But you don't have Schwann cells in your spinal cord, so you don't get that. Yeah. Ooh, his foot got caught in the stirrup? Ooh. Yeah, and that's worse than even a bite wound because you're ripping instead of just slicing. So you're traumatizing more of the nerve to begin with. But depending on how good the surgeon is, that can, if it's pieced together well, it can regrow. So I'm in a Facebook group for biology teachers, and one of them's kid, 1-year-old, got his finger stuck in the door and sheared almost completely off. Now, she's a biologist, right? So she's like, what are the chances that's going to grow back? So she posts on Facebook, what are the chances this is going to grow back? And about 5 people responded, oh, that happened to my kid, grew right back. So, yeah, little tips in babies particularly can, in fact, grow back, regenerate. But nerves will grow back eventually. Now, if the surgeon, and particularly with a twisting wound, if you don't line those nerves up right, you understand that these nerves are physically wired all the way to your brain, right? So if you hook them up wrong, that's like getting wires crossed. So I have a friend who had facial trauma. He had a bike accident, really cut up his face. And when they patched them all up, they said, not so sure about those nerves. There's a lot of damage. They'll probably grow back eventually, but don't know for sure how it's gonna rewire itself. So when it reconnected, the nerves that controlled his salivary glands grew branches to his sweat glands in this right cheek. What does that mean? When do you salivate? Every time you eat. So every time he eats, what else does he do? His right cheek sweats like crazy. He just has to sit there with a napkin on his face. Sweats like salivation. So I had a student tell me when she had knee damage and the surgeon crossed the right or the medial and lateral nerves, sensory nerves. I said, did your brain ever figure it out? She said, nope. Generally, you sort of accommodate to that. But she said, if the lateral side of my knee itches, I have to scratch the medial side to get it. So those nerves will grow back, but there's no guarantee that they know where they're going. The Schwann cells will literally form little growth tunnels to guide them. But sometimes, especially with trauma that just kind of mangles everything, there's no way to know for sure. Okay, so we talked about astrocytes, we talked about oligodendrocytes and Schwann cells. Those are your myelinating guys. The ependymal cells, we'll talk more about when we learn the hollow spaces of the brain. But the microglia I want to mention, these are cells of your immune system. These are the immune cells of your brain. And they are related to macrophages. So what do macrophages do? They eat stuff. So what do microglial cells do? Same thing. They will travel to a site of trauma or infection or disease within your brain, and they just start eating the blood clot or the damaged tissue or whatever. They just eat away. Now, forensically, this is very useful information. If you have trauma to the brain that causes an immediate fatality, the brain's dead, the body's dead, those microglia die, okay? But if you have someone who dies hours to days after the trauma, then the microglia have actually had time to migrate to the site of damage. Now, we're going to wait till the person is dead before we autopsy them. That's just the polite way to do that, don't you think? But if you can take histological sections of the traumatized part of the brain, look for microglial cells. What does it mean if you see them? They lived for at least a while. Whereas if you see no microglial migration at all, then you know that death was practically instantaneous. It's just one of the ways that we tell. Okay? So you understand your brain is an immune privileged site. You have what we call the blood-brain barrier that prevents many things from getting in. But once an infectious organism breaches that, or if you have a stroke that causes, or trauma that causes that, you do have immune cells in your brain that are going to help fight infection or help clean up so that the astrocytes can cleanly form new scar tissue. Okay? All right, so unless you have questions tomorrow, we're done with chapter 12. Okay? Today's Tuesday. We have one day to do brain and spinal cord. And your homework assignment, should you choose to accept it, is to do the chapter 13-14 chapter summary. That will lay the groundwork for what we have to do tomorrow. We have to get through the entire anatomy of the nervous system in one day. So that on Thursday we have autonomic. I know I didn't ask you to do anything today, so if you have your names on the sheet, that will at least give me an attendance record. That would help. All right, see you tomorrow.